take a second to thank the organizers, especially Yang Zhen, for putting this conference together and for taking such good care of us and putting together a scientific program that has just really been top-notch. So thank you to the organizers, yes. So with this slide in the message here, I know I'm sort of preaching to the choir because we're all here because we believe this message, but I really loved this interesting definition of cancer that appeared in an article recently in Time magazine. It says cancer is an intricate and potentially lethal collaboration of genes gone awry, of growth inhibitors gone missing, of hormones and epigenomes changing, and rogue cells breaking free. It works as one great armed force, tacking by the equivalent of land, sea, and in stealth. And we think we're going to cure it with what? A lab-coated sniper? So the real message is that we need a team-based approach. There probably will not be a hero single scientist that comes up with something that can cure all 200 plus types of cancer. So we're all here working towards that goal of building collaborative team-based approaches. And one of the reasons we need these kinds of approaches is because cancer really is a complex dynamical system. Yep?
Sorry, if you're using the microphone, it doesn't sound like...
Put it higher. It's on. I can't. You can hear it. I'm sorry. I'll try to talk even louder. Okay. Oh, okay. So cancer is a complex dynamical system that really scales all levels of biological organization, right? So you can think about things acting at every single biological scale, and you can think about all of the different types of mathematics that might be used to answer questions at each of those individual scales. Or, like a lot of research is aimed at today, is trying to link these different scales, trying to say something about how they're coordinated and how they are connected. So the things I typically look at are at the level of intracellular signaling networks, connecting that up to single-cell decisions, and then moving that on to population-level observables that we can make, like tumor volume versus time, tumor reduction, microvessel density, those kinds of things. But with the drivers really being the molecular-level pieces of that. And I apply that idea to mathematical models of tumor angiogenesis. So I don't think I need to go into too much detail about what angiogenesis is, but of course it's the formation of blood vessels from the existing vasculature. Just a quick note about why it's different in tumors than it is when it happens everywhere else in the body. In tumors, it's one of the only situations where angiogenesis is sustained. It can be prolonged in other pathologies, but in cancer, every increase in tissue mass is typically compensated for by an increase in vasculature. So the tumor ensures itself adequate nutrient supply and removal of waste capabilities by sustaining the angiogenic cascade. When it comes to angiogenesis, there are both cellular and molecular pieces, as well as tissue-level pieces that are important. So let's talk about the cellular molecular bits first. So, of course, there's some tumor, avascular, initially growing somewhere in space, some nearby blood vessel. The tumor cells in the center aren't so happy. They begin secreting not just VEGF, and one of the themes of my talk is moving beyond VEGF in angiogenesis research. VEGF is certainly the most well-studied angiogenic stimulator, but tumors actually secrete VEGF and a whole host of other things that must diffuse and penetrate through the stroma, find its way to an existing blood vessel. And it's really not gradients set up across this tissue length that stimulate processes associated with angiogenesis. Rather, it is the binding of VEGF to cell surface receptors on endothelial cells. They bind to these receptors, dimerize these receptors, and then initiate an entire signaling cascade inside that cell that enables these endothelial cells to survive the harsh conditions outside the protection of a blood vessel, to migrate across the interstitial space and traverse the extracellular matrix and get to the tumor to form these new networks of blood vessels. So that's sort of the molecular and cellular components. And you heard me hint at sort of the tissue level piece. So with this cartoon, I want to just highlight that tissue level piece of the extracellular matrix, here noted by these green little fibers. And I also want to highlight a few aspects of the dynamic of the cells crawling and moving. So extracellular matrix is a main component of the stroma, a fibrous network that cells have to traverse. They use these as movement cues, these fibers as movement cues, and they crawl along these fibers in order to make their way to a tumor site. I'm also showing some of the cellular processes that go on. There can be cell division along a developing sprout, and then there is typically a migrating tip cell with a different morphology than the connected sprout cells that is leading the pack, so to speak. It's extending protrusions, crawling along fibers, helping it find its way, helping the whole structure find its way to a tumor. So there are molecular pieces, cellular pieces, and then these tissue level pieces associated with tumor angiogenesis. So the initial reason for all of the interest in angiogenesis was, of course, anti-angiogenesis. So the goal was to sort of stop this process from happening somehow. The idea, of course, being if we could stop the vascular supply, we should be able to reduce the tumor size. And there have been a number of mechanisms aimed at doing so. So you could try to stop the tumor from secreting this host of angiogenic stimulators. If you can't do that, you could try to stop that group of stimulators from doing its job. You could try to neutralize them before they get to the existing parent vessel. And most people know about Avastin, which is an antibody against VEGF. It acts at this level, right? It tries to neutralize VEGF before it can have an effect. If you can't do that, you could try to inhibit the receptor. So we saw that VEGF has to bind and dimerize receptors on these cells in order to stimulate the activation of those cells. So you could try to stop the receptor activation of endothelial cells. A couple other things you could try to do that are kind of emerging in the field of anti-angiogenesis. So we're trying to move beyond VEGF, right? So we're looking for some VEGF-independent anti-angiogenic targets. So some emerging things that are coming up are inhibiting endothelial cell survival. So when endothelial cells leave the protection of a vessel and begin crawling out in sort of free space, they have to upregulate a lot of survival mechanisms in order to be able to survive those conditions. So you could try to inhibit those survival mechanisms. And then once it starts developing a sprout, that sprout has to mature and stabilize in order to support blood flow. So you could actually try to stop that process as well. Yes.
Yeah, three processes are there at the moment that target each of the three, because you mentioned only about.
Yeah, so good question. So yes, there are FDA-approved drugs that act here, Avastin. There are some FDA-approved drugs that act sort of at this level. And one is approved for kidney cancer and for a couple other cancers that are in advanced stage that act at this level. A lot of these kinds of drugs down here are in clinical trials but have not been FDA approved. Okay. All right. So given all of that, all of the biology of angiogenesis and what we would like to use our knowledge for, you could either use mathematical modeling to try to say something about the basic science of angiogenesis, or you could use mathematical modeling to try to say something translational, right? How do you do something that could actually be clinically useful? And so I've been doing work in both of these arenas, looking at modeling endothelial cell targeted anti-angiogenic strategies that move beyond VEGF, looking at some of these new ones. And when we do this, Ursula and I had an interesting conversation yesterday about when do you need a PDE and when don't you need a PDE, right? So when we do this, the kinds of data I get is tumor volume versus time, blood vessel composition versus time, and we typically use ordinary differential equations, sometimes delay equations, those kinds of things to model that. But when you're looking at the basic science question and you really want to ask questions about the biochemistry and the biophysics, and you're looking at things that are moving in space, partial differential equations or hybrid approaches can be also useful in that situation. So today I'm going to focus on the basic science questions of combining biochemistry and biophysics to understand some of the mechanisms associated with angiogenesis. All right, so what kinds of questions do we want to use modeling to answer when we're looking at the basic science questions? Well, we want to know what factors influence cell phenotype distribution in a developing sprout. So not all cells in the sprout behave the same way, but they're all important. So how do we know when we're going to have a quiescent cell, one that's not dividing, a proliferating cell, or a tip cell that actually has that different morphology? And how does the dynamic variation among those phenotypes, how can we characterize that using mathematical modeling? We also want to ask questions about how do these tip cells really behave? How can they polarize themselves in response to chemical gradients of stimulators of VEGF and other chemokines that are bound to their surfaces? So we're not talking about gradients set up over the domain of a tissue. We're talking about gradients set up over the length of the cell. So how can we incorporate that kind of thing into models? And how do these endothelial cells integrate the various biochemical cues with the biomechanical cues they receive from all of these different sources and determine how they're going to move? So those are the kinds of questions we're interested in. All right, so the first type of mathematical model I'm going to show you is it uses a hybrid approach, and we're really aiming at that question of can we include some of these molecular level dynamics? And even though this is the basic science talk, why are we trying to include these molecular level dynamics? Our eye is always towards molecular therapeutics. They are targeting these particular molecular dynamics. So if we can include them correctly in a mathematical model, we should be able to have a better estimation of therapeutic outcome when we move to translational. So what is a hybrid model and why go with that approach? I define a hybrid dynamical system as one that has components that are both continuous and discrete. So a system that has components that can both flow or diffuse, like these chemicals like VEGF, or they might actually have components that actually jump or move more discretely, like endothelial cells. So we talked about that specialized tip cell in my cartoon. Here's an actual image of such a cell protruding lamellipodia and trying to crawl in space. And that's why we want to use a hybrid approach, so we can have diffusion of VEGF, but also the crawling or discrete motion of individual endothelial cells. Another reason is because when you add things like division of these cells along the stalk, that's hard to capture with just a continuous approach. All right, so now we're just going to focus on the molecular dynamics with a very simple goal. We're going to want to see if we can capture the binding of VEGF by a sprout tip cell first, and measure the activation of that tip cell based on the surface receptors that cell has, and quantify the chemotactic response of that cell based on this binding of VEGF. And this is kind of different than what's out there in the literature because we're building from the molecular level model, and we want to capture that polarization across a length of a cell instead of across the length of a full tissue. Okay? So here's a schematic diagram of the model. We're going to start with one end of a 2D domain that has a parent vessel outside of our region of interest that can produce single endothelial cells that are going to traverse the domain. We'll have a source of our chemoattractant, VEGF, on the other end of our domain, and it will diffuse out and set up a gradient. We're going to just look at one tip cell for now, so this is just a model for what happens with one cell, and then we'll make that cell the leader of a vessel and show you what happens in a few minutes. So we will center this cell at some point in space. It will have receptors on each cell face. The VEGF that has been secreted by the tumor will bind to those receptors. Based on that binding, the cell will choose a direction to elongate, so it's going to elongate in the direction of the greatest increase in bound receptor, and it's going to move in that direction. So it will move one grid point after the binding. So that's the idea. So how are we going to model that? Well, well, there's lots of ways we could do this, but we decided to start with a master equation for cell movement that starts off from the theory of random walks. So if I were going to do an unbiased random walk, of course, in 2D, I would say I would start at some position, and I would move a certain length delta x every time step delta t. And if it's not biased, I would say that the probability of moving left, right, up, and down would always be a fourth. And so I could write the probability that the walkers, or my cells, would be at position x and y at time t quite easily. We want, of course, our cells are biased by a lot of things. They're biased by everything in the microenvironment, especially these vascular endothelial cell growth factor molecules. So we need to modify that equation to be a biased random walk. So now we're going to write the rate of change of the probability that a cell is at a particular position in M based on transition probabilities for moving in each direction. This is a horizontal jump to the right, horizontal jump to the left, vertical jump up, vertical jump down, and a cell could also choose to stay where it is. Okay. All right, so then we have this equation, and we're going to make some assumptions that make sure that we get back to a standard equation when we take the continuous limit. So one of our big assumptions is that the decision of where to move is independent of the decision of when to move, and we can satisfy that assumption by choosing a simple jump process that makes it true. And Hans can verify that that can be done. And if we pass to the continuous limit, we get something that looks like a traditional diffusion chemotaxis type equations. But when we solve this numerically, we're actually going to stick with the discrete version, so we never have to solve the continuous equation. We just want to make sure that we actually got movement patterns that look like they should in the continuous setting. All right. Another thing we want to do that is kind of important is change up the traditional thinking about chemotactic sensitivity. So evidence points towards the existence of a minimum threshold of the stimulus that's required to activate these receptors, all right, to start moving. And the amount of that chemical or chemokine required to desensitize a cell depends on how many cells are there, right? If you have a small amount of VEGF and a small amount of cells, you might be able to desensitize it, but if you have more cells, you will not be able to desensitize that group of cells with the same amount of chemokine. So we want to capture some of those things, and the biggest thing we want to capture is that in eukaryotic cells, it has been reported that in order to polarize a cell, you can do that with gradients of chemical of about 2%, just 2% across the length of a cell. So we don't need this big gradient across the tissue. We should be able to polarize the cell with small amounts of bound chemokine across its length. All right, so we're going to make some biologically more reasonable assumptions to try to capture some of these things about our chemotactic sensitivity. All right, so when we do that, now our equation would, instead of having free VEGF be the driving force for our chemotactic motion, we need to have bound VEGF per cell face be the driving force of our motion. So we have a velocity imparted to our cells by the bound VEGF, and the gradient of the bound VEGF across the cell faces gives us the direction of polarization. And for our, yes, sorry. Which one? Yes, dp is constant. Yeah, thank you. So we choose a function for our chemotactic sensitivity that's a little different than the traditional use of a constant or a receptor law. We choose a functional form that has chemotactic sensitivity being 0 when there is no chemokine, which sort of makes sense. And it also goes to 0 as my concentration of bound receptors gets very large. That's the desensitization portion. And then there's a region where we have high sensitivity, and that region is sort of centered around the 2% polarization mark that we talked about. All right, so when we run simulations of this, some of the things that we can get out of this just for the single cell is looking at how fast or how long it takes the cell to migrate across the domain. If we give it different amounts of free VEGF in the medium. Even though we're measuring this based on how much free VEGF we put it as available, remember it's the bound VEGF that's causing the cells to move. So we see at low concentrations of VEGF, we don't get enough binding to sensitize the cells, and it takes a long time. The same thing happens if we give too much VEGF. There's too much bound to the cell surface, and it again takes the cell a long time to traverse the domain. But then if we give optimal doses, smaller doses, we get the fastest migration times across the domain. So here, so error bars are for, yeah, so, I left out some details. So when we simulate that discrete version of the model, we sort of follow the Chaplin-Anderson approach, where we split the domain and we choose a random number and decide which probability up, down, left, right for the cell to move. So the error bars are for several runs. Okay, so here's just a picture if we just follow that cell and look where it traveled under those three conditions. So this is with a small amount of VEGF when it took a long time to traverse the domain. We see that's because the cell doesn't really have a great sense of its direction of where it should go, and it moves around a lot before eventually getting there. The same thing happens, and maybe even to a worse degree, if we have too much VEGF. But if we put in those smaller doses near the optimum, we see the cell almost makes a straight line path across the domain. This side just shows those movement probabilities, and we see that when we're near the optimum, we're of course moving to the right with the highest probability, and not so if we have too much or too little VEGF. And one thing about this model is that we can actually look at the fractional occupancy of the receptors. So we can see how many receptors were bound and what the occupancy was for each one of these movement patterns. All right, so now we kind of understand how we can do this for a single cell, but we want that single cell to act like a tip cell guiding a sprout. So we need to add a few extra assumptions. We'll keep the domain looking exactly the same, but now we're going to say that we have 4 little initial sprouts positioned along the y-axis, and we're going to track the movement of the tip cell as they pull and drag a developing sprout behind them, and kind of computationally costly, but we're going to keep track of every site that the cell visits. Because it's got cells behind it, we can't have backtracking, right? So we make sure the cell is not able to backtrack. So we kind of reinforce the random walk so that movement backwards is undesirable. So here is what would happen if we let that tip cell be the leader of a vessel. We can look at what happens if there is too little VEGF. We get very stunted growth of our sprouts. We can look at what happens if we get too much VEGF, and this is a situation where cells are just moving around and sort of are desensitized. The reason we have such a thick network of vessels is because the amount of VEGF also plays a role in whether or not the vessels will branch and anastomose. And then at the optimal concentration, we see the most streamlined group of vessels approaching the other end of the domain. The domain, which is a tumor. Yeah. So rules for the following cells. So they just follow exactly the path of the tip cell. If they, and we have some other rules about when the tip cells should branch and when they should anastomose, but the trailing cells really are passive, and they don't do anything but follow the leading cell. We're going to move to models where that's not true, because that is a big assumption here. So yeah, that's the next step, actually. Yeah. Yep, that's exactly right. So yeah, so I mean, there was a concentration right around here that triggered the leading cell to cross the domain the fastest, and we called that the optimal external VEGF concentration. It's actually the optimal speed of getting across. Mm-hmm. Yes. Alrighty. So there are some pros and cons of this model, and Hans just highlighted one. Some good things are that we were able to include some of this molecular level detail and show how that can help guide the motion of a tip cell. We could capture the activation and polarization of that special tip cell, but this model does not have a realistic representation of cell matrix interactions and the mechanics of cell motion, and it also made a big assumption about those trailing cells. They were passive, they really didn't contribute anything to the sprout, and that's not true in the real situation. All right, so we want to move to models that can maybe say something about cell ECM interactions without losing some of the things that we gained with the molecular level detail. So we're going to use a slightly different approach. I kind of set this up already. Why do we need this approach? Well, the big reason is we don't want all the cells in the sprout to be inactive and sort of passive contributors. We know that they actually do a lot of things, so we want to include them. And then in that approach we just talked about, we had to make rules about when will branching occur, when will anastomosis occur. And in vivo, right, those things just happen, right? So hopefully we could get those processes to occur naturally without a priori prescribing rules for that. So we're going to look at a slightly different approach. It's still hybrid in that there are discrete components, if you will, and continuous components like VEGF, but we're going to use a cellular POTS model to describe what happens with the endothelial cells. The cellular POTS model is a lattice-based Monte Carlo method where we give cells a finite volume but a deformable shape and let them interact with each other through their surfaces, through a surface or adhesion energy. So what this picture shows, and then the system runs to minimize total energy. So cells have a type, so E represents an endothelial cell, and T represents a stromal cell, M is an extracellular matrix fiber, and F is the fluid, interstitial fluid, so we have 4 types. And each cell has a unique identifying number, so E4, everywhere you see an E4, that's all part of one cell, and E3 is all part of one cell. And so we assign that to every spot that a cell occupies. We write an equation for the total energy. So we have energy associated with interactions of cells with each other, cells of the same or different type. And then we have some energy required for cell growth. So cells have some initial starting volume and some target volume. And to grow beyond that target volume costs you energy. Okay? So then we let the system run based on an equation for total energy. Here's what that would look like. So we do continuous probabilistic updates of the cell's boundaries. So for example, we could choose a random site and switch its ID to one of its neighbors. That represents an interaction. Sort of if 3 is an endothelial cell and 2 is a matrix fiber, we could choose a site on that endothelial cell and see how it would interact with a component of the extracellular matrix. Sorry. There we go. So we change it to one of its unlike neighbors, and we calculate the energy, change in energy that switch made, and then we accept it if the energy is reduced. If the energy is increased, we accept it with a probability, a Boltzmann probability. Okay? And we let the system run until energy is minimized. Another interesting thing about this, remember we said we wanted to have a more realistic representation of cell matrix interactions. So we actually wanted to try to put in a realistic representation of the extracellular matrix. So here on your left-hand side is an image of extracellular matrix fibers, pretty much a pretty zoomed in image of extracellular matrix fibers that cover about 37% of the stroma. And what we did to try to mimic this was randomly distribute fiber bundles at orientations between 0 and 180 degrees throughout our space until about 37% was occupied. And those are going to be what cells use as movement cues. All right, so here's our domain. Just like before, a parent vessel on the outside of a domain, a tumor on the other side, setting up a gradient of VEGF. I forgot to mention, we haven't lost everything we said about binding of VEGF. All of that still happens with a continuous diffusion equation for VEGF fusion and binding when there is an endothelial cell present. All right. So here is an example of a simulation of this model. Remember, blue are stromal cells, red are endothelial cells, light blue is fluid, and yellow are extracellular matrix fibers. What you see is these cells really are crawling along the fibrous structure. Another thing you see is when two sprouts came together, they formed a closed loop and continue to move. All of this without any rules. There was no rules for branching. There was no rules for anastomosis. None of that was prescribed a priori, but it was an emerging phenomena based on the minimum energy. All right, so a couple other things you can do with this model is look at different isoforms of EGF and how they would affect sprout development. So I'm sorry, go ahead.
So I mean, I read that of course there is a difference between normal angiogenesis and tumor angiogenesis.
Sure.
Because the tumor is like more violent, more sort of, they call it abnormal.
Yeah.
So is this a simulation would look different? It would just be simulating regular angiogenesis?
Good question, and the answer is no, not yet, because I so far have modeled the early events in angiogenesis. When it starts really getting tricky and abnormal happens a little bit later on. It's the way that these vessels mature and stabilize. They're leaky, and so all of that happens a little bit after this model's timescale of interest. Yeah. All right, so you can see that if you have an isoform that binds to extracellular matrix, that's this figure, and you have a steep gradient of the EGF, you get these thin sprouts, like a picture here of the EGF 164. And if you have a more soluble isoform of the EGF that sort of sits around in the extracellular matrix, you get these fat, plump vessels that move a lot slower, and you get a shallow gradient of EGF. So we just use it to check a few things like that. Since extracellular matrix is one of the things we added, we wanted to see how the fraction of matrix fibers affected the extension speed. So we looked to see if we had a denser structure for the endothelial cells to traverse versus a sparser structure, how would that affect the times? And we also see that there is sort of a region where there is optimal, fastest extension speeds for intermediate levels of fiber densities. So if there's not enough ECM, the cells have nothing to crawl on. If there's too much, then the cells, can't traverse that domain. So there's some pros and cons of this one. It was nice to include the extracellular matrix. It kind of produced some pretty pictures. And we were able to track all the cells in the sprout. The problem with that is we tracked every single shape and morphological change of every single cell in a developing sprout. And it became very computationally expensive, especially if you ever thought about wanting to go to 3D. It would be very difficult. Also, with the eye towards molecular therapeutics, if we wanted to add more molecular dynamics, more than what we had, that would also be a challenge to say anything about real treatment strategies with this model.
Okay, in addition to this video, she investigated the effect of the extended speed, the function of geometric coordination of the fiber.
No, actually we didn't, but that's a good idea. Yeah. Yeah, so baseline was 37%, and then we went up and down from that. But no, we didn't look at any fiber orientation. That's actually a good idea, because if the fibers are already aligned for the cells, right, cells are going to move faster. If the cells have to do some work, I would imagine it would be slower. Yeah, good question. Okay, so let's do our last little modeling venture. Let's try to do all of this, keep all of this stuff, but lose the stuff that's making it so computationally costly. So we want to still have all the cell phenotypes and talk about the active role of all of the cells in the stalk without modeling every single morphological movement and change of those cells. We just want to model their effect on sprout development. Okay. All right, so we are going to have a model for the tip cell and a model for stalk cells so that we still are tracking every cell in the sprout. We just don't have to know how they're changing in space and time. So we're going to say the vessels are made up of both kinds of cells. Stalk cells are still active participants. They bind VEGF as well as angiopoietins. Angiopoietins are the mediators of maturation and stabilization. So we're trying to get to that point where we're having real differences between normal and abnormal vessel growth by modeling the maturation process, which is where a lot of things can go wrong. Once the mass is doubled in a stalk cell, we actually have them divide into two cells and redistribute in space. We have a non-proliferative mechanism for sprout extension, so we can actually have cells from the parent vessel migrate along an endothelial cell sprout and take its position at the leading edge. We use discrete points to track all of the cells in the vessel, and on each one of these cells in the stalk, we look at its sort of quiescent level, or how mature it is. We sort of equate maturation with quiescence, so the immature cells are the ones dividing and leading, and the mature cells are the ones that are stabilizing the vessel and have stopped dividing, and we track all of that along the sprout. So that's what we do with the stalk. For the tip, we sort of use a mechanical model to say that the tip has two parts. It has the parts that are protruding and using the fibers, and then it has its main body. And in the vessel, the main body is sort of attached to the other cells behind it. So we could sort of think of this as kind of a tethered spring. So the back of the cell is sort of tethered to everyone else, but the front of the spring is free to kind of stretch and retract. And if we do that, we can model it with a spring dashpot system where we write a force balance equation for the elastic, internal and viscous forces, the drag forces, and the protrusion force is the force that the cells use to extend itself, is driven by chemotaxis. So I'm calling that a chemotactic protrusion force. All right. So we do all of that, and just a reminder, we're actually adding more molecular dynamics here. So we have all of the VEGF stuff that we talked about earlier, but now we're adding this maturation component with the angiopoietins and their natural receptor. So we're building up to more and more molecularly realistic models of angiogenesis. So the algorithm for this one is, first we solve the tip cell elongation equation, and we let it reach its steady state. Then we solve the chemical equations and figure out how mature cells are and if they're ready to divide by whether or not their mass is doubled. If there has been a division event, that gives my tip cell a little push. It can move forward and extend. If not, it can only extend to its steady state length. If two vessels interact in space, they fuse and anastomose, like we've said before. And if it has enough mass and VEGF, it can branch into other cells. And so we repeat this process in order to get a dynamic picture of the changes in them. So here's an example of, so we used a 2D domain where we have a tumor source in the center. So this is like corneal angiogenesis, a tumor source in the center with blood vessels, parent vessels along the outside. And we watch what happens as these vessels extend in space. So your panel on your left-hand side is at day 4, and on your right-hand side here is day 7. So we're just comparing the length of our vessels and their extension speed to experimental data, and we use that data to sort of calibrate some of our parameters and get good fits. Over here you see the maturation level, so red being the most mature. So on day 4, almost all cells are immature and are dividing, but by 7 you see maturation has started to happen near the base of the parent vessel and is spreading out from there. And the last two sets of graphs are looking at the angiopoietins, which govern the stabilization and maturation and how those densities vary along the sprouts. One thing we used this model for was to look at the proliferation region. So there was some controversy about where that would be in a developing sprout, so we just looked at our model to see where that would be. So on your left-hand side, this is the percent of cells that are dividing within 0.5 millimeter of the tip. So within half a millimeter of the tip, and this is time in days. So we see that almost all the proliferation is occurring right behind the tip. And this is just another way of looking at that, the number of dividing cells as a function of the distance to the tip. So distance to the tip is very small. On day 4, you have almost all the cells there, a large percentage of them dividing, and then that stabilizes and spreads out and reduces quite a lot as time progresses on day 7. All right, so we use this model to look at a couple different, to train the model with a couple different experiments on treatment. So this is irradiating the cells so they can't divide. The red represents, the red line is a simulation, and the couple little circles are the only two experimental data, or the couple of experimental data points we had for what happens if there is no treatment, looking at how far you can move into the domain versus time. And then if you irradiate the cells with different doses, you get that extension significantly reduced, and those are the model simulations of that. And then these two graphs just look at what happened if we remove certain elements of the model. So if we remove branching, that's no splitting, we get the extension to be too fast. And if we remove maturation and let all the cells stay immature and proliferate, we also get very rapid extension. So those two mechanisms are actually really important to help get the experimentally reserved control case. So this is just a picture of the 2000 RADS experiment. So this blue line, this is just a picture of what happens looking at how far the cells extended into the vessel. This is looking at how mature they are. So actually, we get these stunted little vessels that are all made up of fully mature cells. Nothing else is dividing, so nothing else is extending. And these are pictures of the angiopoietins on those cells, you see, which govern that maturation function. All right, just about done. One other thing they did experimentally that we just wanted to check that we could sort of capture at least qualitatively with the model was they sort of irradiated a certain percent of the cells first to make sure a certain percent of the cells could not divide, and watch what happened in time. So this blue one is the triangles are day 4 and the circles are day 7, when a certain fraction of those cells were initially quiescent. So we did the same experiment, and we see that if we want to compare to the experimental data, we would look at the steady state length as a function of how many cells were initially quiescent, and we get a very similar trend to this data, to what they saw experimentally. We also tracked other things like the percent of cells that were quiescent as a function of the initial amount, and eventually every cell is quiescent. We saw that in the last figure, all cells were mature, and we also looked at how the length of the vessels as we varied that as well. All right, so let's just summarize really quickly. So we kind of used this particular model to get rid of some of the computational issues we had with the others and still advance our understanding at the molecular level, and to sort of tease out some of the mysteries around elongation, proliferation, maturation, get us closer to saying something about the difference between tumor angiogenesis and regular angiogenesis, and looking at the interactions among events in a way that really hadn't been done before. So those are just a few of the things we found. I already went over those. So here's how everything kind of evolved. So angiogenesis and modeling of angiogenesis is not a new field, right? In fact, when you saw that title, you're probably like, oh my God, another angiogenesis model. That's how. But things have really evolved and moved well beyond the old version of angiogenesis models that you probably were thinking of. And just in this talk, we moved from hybrid models following single cells, but including molecular dynamics, to hybrid and cell-based models tracking every single cell in the sprout and the molecular dynamics. That gets us closer to being able to say something real about molecular therapeutics aimed at targeting blood vessels. A couple of ongoing research projects. So like we said, we're trying to move beyond VEGF and anti-VEGF as a therapeutic strategy. So we're looking at drugs that move from highly specific to broad range inhibition of survival. So we've got some models of that. The model we just showed you that had angiopoietins and maturation can be used to simulate drugs that target that process, and there are several that are currently in clinical trials, so we're running simulations to do that. And then the story I just told you, and the story you're probably most familiar with, is the one-way story of tumors secrete things and endothelial cells respond to those things. Really, the communication is a two-way street, right? So endothelial cells secrete things that the tumor responds to. So we want to actually build in the two-way communication pathways between endothelial cells and tumor cells, and look at targeting both at the same time, maybe even determining which type of cellular inhibition is most important for the overall therapeutic effects. Really, what's on the horizon is using these molecularly driven models of blood vessel formation to connect to three-dimensional models of vascular tumor growth. So these are simulations that came from the Lohengruber group and Christini group, looking at, so the squigglies are old version of angiogenesis models that are driving the growth of these tumors. What we want to do is put in these molecular level angiogenesis models to drive growth of tumors so that we can say something about combination treatment strategies that may use the vasculature as a delivery mechanism and attack the blood vessels at the same time. So sort of looking at all the kinds of treatments that Ursula mentioned in her talk. So that's on the horizon. All right, a quick thank you to the people who helped with this stuff. My experimental collaborators, Jacques Noor, computational collaborator, Yi Zhang, graduate students and postdocs, and funding agencies have all contributed to this. So thanks for your attention this morning.